Okay, hello everyone. Welcome to VSPG. Today we're hearing from Dr. Cecilia Sanders, who's now at Johns Hopkins University, on phosphorites under the microscope, microbial ecology, and the phosphorus cycle across the Precambrian-Cambrian boundary. Okay, Andre, could you please go ahead and introduce Cecilia for us?
Yeah, it's a pleasure to introduce Cecilia. Cecilia did her bachelor degree in Harvard University and started working on planetary geology. She followed with master and PhD at Caltech, where she switched gears to sedimentology and sedimentary geochemistry, I guess working on clumped isotopes and sulfur isotopes. And when she followed with a postdoc from Johns Hopkins University, where she is now. And in summer, she is switching for a 1 year postdoc at Smithsonian National University and will start in summer 2025 at University of Maryland. So with this, it's a pleasure to pass it to you, Cecilia, and take it over.
All right, thanks very much, Alex and Andre. And hello, everyone. Thank you so much for inviting me to speak. I've really enjoyed this whole seminar series, learned a lot, and I've really appreciated having this forum for the discussion and sharing of ideas. So yes, thank you very much to the organizers. I want to speak to the group today about the work that I've been pursuing during and since my dissertation, diving into the geobiological record as preserved in phosphorites. So I'm very eager to get y'all's thoughts on some of the ideas and methodologies, especially with respect to the preliminary data that I've yet to publish from my postdoc at Johns Hopkins. So let's get into it. I'll go ahead and start with an introduction to the study of phosphorites and their formation, a process called phosphogenesis. This will be a brief review of all the reasons why phosphorites should be of interest to students of environmental and ecological history, particularly across the Precambrian-Cambrian boundary. Then I'll go through a comparative synthesis of my and many, many others' geological and geochemical investigations of several different phosphorite deposits from the Precambrian-Cambrian boundary as exposed around the world. And I'll finish up comparing what we see in the geologic record to models of microbially mediated phosphogenesis based on modern systems. And we'll finish out sort of teasing this emerging hypothesis we have about the significance of phosphorites for ecological evolution during this Precambrian-Cambrian transition. So let us get after it with phosphorites, an introduction. So what are phosphorites in the first place? Phosphorites are phosphate-rich sedimentary deposits in which phosphate is typically concentrated in phosphatic minerals, so typically apatites, calcium phosphates, which can contain a wide range of substituents. There are many different types of phosphorites occurring throughout the sedimentary record, from terrestrial coprolites to lacustrine nodules to marine reefs. So why are phosphorites in particular of interest to geobiology? Firstly, and perhaps most importantly, they contain phosphorus. Phosphorus is an essential nutrient and component of biomass, and at many times and in many settings, it is often the limiting nutrient for biomass production. It's in cell walls and membranes, nucleic acids, and its addition or removal to and from various enzymes in the cell and its membrane is responsible for chemical energy transfer from an organism's central metabolic reaction network to its essential life functions. So because of its use in metabolism, its cycling through biological and abiological pools is often coupled to the cycling of other essential nutrients and metabolites, including carbon, sulfur, and iron, and other important metals. I've included this little plot on the right. This is just from the original paper that discusses what we now call the Redfield ratio. This describes the relative abundance of carbon, oxygen, nitrogen, and phosphorus in average biomass, emphasizing that this is an important element for the history of life. So phosphorites represent the burial of tremendous quantities of phosphorus, this limiting nutrient. And so the formation of phosphorites must correspond to major perturbations in the production and cycling of biomass itself. Another reason to take an interest in phosphorite formation is that the permineralization of organic structures by phosphate minerals can preserve subcellular detail with high fidelity. This is due to the thermodynamics and kinetics of apatite crystal nucleation and growth, as well as the role of organic matter itself in sourcing and templating phosphate, and the resistance of apatite crystals to fabric destructive recrystallization under burial conditions over geologic timescales. So some of the most rich microbial lagerstätte are preserved in phosphorites, such as those of the famous Ediacaran and formation in China, which is demonstrated by the exquisite preservation of these cells in the process of dividing. These images are from Xiuhai Xiao and Andy Knowles' 1999 paper about this formation. But wait, there's more. The mineralization of phosphate requires the right window of physical and chemical conditions to happen, meaning that the existence of sedimentary phosphorite itself is a kind of environmental dipstick constraining primary and early diagenetic chemistry from the time of deposition and cementation. And indeed, the mineral structure of apatite is very accommodating of lots of different ionic substituents. So it incorporates, as it precipitates and grows, contributors to pore water alkalinity, such as sulfate and carbonate, as well as redox-sensitive metals like samarium and other rare earth elements and uranium. So after precipitation, apatite minerals are then fairly resistant to diagenetic resetting. So they provide this fairly stable record of paleoenvironmental conditions. And interestingly enough, the abundance of sedimentary phosphate deposits, over geologic time seems to correspond closely with periods of major environmental and ecological change, including the Precambrian-Cambrian transition and the Permian mass extinction. Which means that not only do phosphorites provide this record of environmental conditions and biogeochemical cycling of the essential nutrient phosphorus, but those records are at their most robust and widespread during the most geologically interesting periods of Earth history. So, how are these sedimentary phosphorite records actually written, and what controls their spatial and temporal distribution? So this boils down to two components, processes that affect the accumulation of phosphate in the environment and processes that affect the retention and burial of phosphate in the sediments. So what kinds of processes contribute to accumulation in the environment? So it's just kind of a brief overview. I would refer the listener to Tom Laxo, Eric Sperling, Dave Johnston, and Andy Knowles' paper from 2020, which makes a very compelling argument that phosphate accumulation in the geologic past, and particularly at the Precambrian-Cambrian boundary, resulted from delivery of phosphate from deep water reservoirs of nutrients derived from remineralized organic matter, delivery of those nutrients to the surface ocean due to a combination of geography, upwelling, and post-glacial overturn of the oceans. So this is overturn as cold glacial meltwater at the surface sinks and displaces deep water. That deep water's got to go somewhere, so it brings all these nutrients up into the surface ocean, which would directly lead to an increase in biomass production in this post-glacial period. Meanwhile, you have the rise of marine oxygen, which would have directly increased the availability of sulfate, and thus the potential for microbial sulfate reduction of all of this newly generated organic matter, and then the release of excess inorganic phosphate throughout the oceans. Now, of course, another potential driver for phosphate accumulation could be increased input into the global system from detrital apatite weathering off the continents. But Tom Loxo's paper discusses how kind of the timing of the record of orogeny and detrital inorganic p-flux that we have from shales doesn't quite seem to correspond with a record of sedimentary phosphate burial. So perhaps if Eric Sperling or anyone else from that paper happens to be there, maybe we can talk more about that later. But general consensus seems to be that rather than phosphate accumulation corresponding to an increase in inputs from the surface, from weathering off the continents, we think it's an increased influx of phosphate and other nutrients from the oceans. So that is phosphate accumulation. But what about phosphate retention in sediments? What processes control the burial of phosphate as authigenic and early diagenetic apatite? So here is a simple model that is widely accepted for phosphate delivery, retention, and burial in marine sediments. Here's the sediment-water interface. For our purposes, we have relatively oxic waters above and increasingly anoxic waters below that permeable sediment-water interface. So the major mechanism for P delivery to sediments, aside from detrital apatite, is as organically bound phosphorus sinking from the water column or generated by primary producers near the sediment-water interface. This biomass is remineralized via aerobic and anaerobic respiration, and the resulting free phosphate taken up and recycled in the production of yet more biomass. But some of this biomass, rather than being recycled, will be buried below the sediment-water interface. And below the sediment-water interface, where diffusive and advective transport of oxygenated water from above is limited, pore fluids will become increasingly reducing, and the dominant means of remineralization and inorganic phosphorus release will be anaerobic metabolisms, such as bacterial sulfate reduction. This transition from oxic to anoxic is referred to as the chemocline or the redox cline, whose depth below that interface may vary according to various depositional factors like sediment flux or diurnal cycles of biomass production, and so on. Other means of getting phosphate into the sediments is simple advective or diffusive transport of free phosphate from the water column, as well as the burial of detrital iron oxides and oxyhydroxides. These readily absorb and scavenge inorganic phosphate under oxidizing conditions. And then as these iron oxide phosphate complexes are buried below the chemocline, they are reduced and dissociated, and free phosphate is released into the increasingly anoxic pore fluids. Advective and diffusive transport of phosphate from below the chemocline to above the chemocline is thus limited due to scavenging by iron oxides. So this amounts to a sort of iron trap, which helps retain phosphate in pore fluids below that chemocline. So then pore fluids below the chemocline thus accumulate free phosphate from anaerobic respiration of biomass and reduction of these iron oxide phosphate complexes until saturation is reached with respect to phosphate minerals, i.e. apatite. So in this model, some phosphate, there we go. Some phosphate remains to be cycled in the ocean and shallow pore fluids, while some is truly retained in the sediments and buried ultimately as this authigenic or early diagenetic apatite. Now, ostensibly, this should be happening everywhere that there is biomass production and redox stratification in the sediments, but it doesn't. The geologic record, as well as the distribution of modern phosphogenic environments, suggests that phosphogenesis is only happening at certain special horizons in geologic time and only in certain environments. This is probably because even though the conditions presented in this basic model are widespread, there's a lot of variability in the world with respect to the phosphorus content of the biomass delivered to the sediments, the location of the chemocline relative to the sediment-water interface, as well as the availability of other limiting nutrients and of pH and factors affecting the kinetics of appetite, nucleation, and growth. As well as even the availability of iron oxides for adsorption and scavenging of free phosphate. So how did phosphorite deposits and their syn-depositional non-phosphatic sediments vary with respect to these factors across the Precambrian-Cambrian boundary, and at what spatial scales did they vary? So these are the kinds of questions that we're trying to answer in the comparative analysis of phosphorites from the late Neoproterozoic, the Cryogenian, the Ediacaran periods, and the Cambrian period. So we want to compare these with respect to their depositional environments, paleontological potential, and geochemical proxies for local paleo environment, paleo-ecological factors. So this is a relatively old map compiled by Peter Cook and John Shergold, showing the modern exposures of Neoproterozoic and Cambrian phosphorite deposits known as of 1984 when they published this paper. Happily, my advisors of the past 10 years maintain connections with collaborators in several of these locations, namely in Eastern Brazil, Southwestern Mongolia, and Southern Kazakhstan. So with their support, I've been able to measure sections and collect samples through the phosphatic and non-phosphatic facies of these sequences. So in the interest of time, I am going to blow by some of the stratigraphy and regional geology of these formations to focus on characterization of the outcrop and the microscale. So if you have questions about those larger scale data, I will have to refer you to some of the recent literature, or we can discuss afterwards, but I am going to dive into the overview. So the Salitre Formation of Eastern Brazil represents phosphorite and carbonate deposition in a shallow intracratonic sea environment during the Ediacaran period. Phosphatic facies therein include branching digitate stromatolites, which are cemented by cryptocrystalline carbonate-rich fluorapatite, which sometimes called CFA, sometimes called francolite. These stromatolites occur in patch reefs and meter-scale tabular buildups among this dramatically cross-stratified and often oolitic carbonate grainstone. So detailed stratigraphic measurements, outcrop sketches, these contextualize the finer scale observations and thin sections, which altogether reveal that the sedimentary conditions for both the phosphatic and the non-phosphatic rocks of this formation were essentially identical. So, if not depositional environment, location within the geometry of the carbonate platform, if those aren't correlating with the distribution of phosphatic versus non-phosphatic cements, then what does make the difference? What makes the difference between the places where early phosphate mineralization occurred, which is the stromatolites, and where it didn't, which is other stromatolites, as well as the interstitial grainstone. So we compared phosphatic and non-phosphatic facies in thin section via optical microscopy, X-ray fluorescence spectroscopy, SEM, EDS, as well as stable carbon, oxygen, sulfur isotope geochemistry. And here is what we found. So these are some representative images, plane polarized photomicrographs on the right, cross polarized photomicrographs. Sorry, strike that, reverse it. These images show how phosphate occurs in these rocks as cryptocrystalline apatite within and immediately adjacent to digitate stromatolite lamina. It also occurs in rounded intraclasts, but these intraclasts often have internal textures similar to the stromatolites, suggesting that they are formed via the reworking of apatite cemented stromes, or stromatolites, sorry, and their associated nearby crinkly laminations. X-ray fluorescence maps as well, which are shown here, as well as energy dispersive spectroscopy, which I didn't include here. These data reveal no obvious spatial correlation between iron and phosphate in the phosphatic stromatolites. You might expect that if iron oxide burial and this iron trap were responsible for phosphate retention in the sediments, you might expect to see a spatial correlation between iron and phosphorus in the rocks, but we don't. Rather, the distribution of phosphorus seems to be correlated more with the occurrence of sulfur, which itself was associated with diffuse organic matter, presumably derived from the microbial mats that form the stromatolites. This association, where in non-phosphatic stromatolites, you still see some sulfur, you still see iron, you still see these other things, but they're not necessarily enriched in the stromatolites themselves. So if sulfur is so important, maybe we should look at geochemical proxies for biogeochemical sulfur cycling at these highly local scales, comparing non-phosphatic and phosphatic facies. So as it turns out, the sulfur isotope composition of the reduced and oxidized sulfur phases were nearly identical in the phosphatic facies. But in non-phosphatic facies, they differed by on the order of 10 to 20 per mil. So the details of these measurements, how they were done, why they were done the way that they were, also happy to discuss after the fact, or I can refer you to our paper from earlier this year. But suffice to say that there does appear to be this systematic difference in how information about the sulfur cycle has been recorded in the phosphatic versus non-phosphatic facies. Is that a difference in the forming of those records, or a difference in the primary or original sulfur dynamics in these systems? So altogether, these data suggest a scenario wherein the spatial distribution of phosphate mineralization was not controlled by surface conditions, factors like sediment flux or water depth, or even the presence of microorganisms themselves, but rather, nor the availability of iron for phosphate absorption and scavenging in the sediments, but rather as microbial mats, sediments are buried at increasingly deep depths, it seems that there emerged highly local variations in the phosphorus content of biomass and the relative depth of the redox cline, some factor that made phosphorus, that enriched phosphorus or caused it to be retained in one cubic centimeter and not the next cubic centimeter. So it seems plausible that one of the few factors to vary on that kind of spatial scale might be microbial ecology, the composition of the microbial communities living in one versus the other patch of the sediments. By comparison, the phosphorites of Southwestern Mongolia, captured in the Bayangol and the Selangol formations, span the Precambrian-Cambrian boundary, similar to those of Eastern Brazil, but the nature of phosphate mineralization appears to have differed fundamentally in several different ways. So again, I'm going to blow past some of the regional geologic context to focus on finer spatial scales, but suffice to say that though these phosphorites were deposited under somewhat more complicated tectono-sedimentary circumstances compared to Eastern Brazil, the depositional setting was still essentially a carbonate platform or ramp kind of environment, ranging laterally from the relatively wave-swept and energetic to more quiescent deep waters. So phosphate occurs in the biangle and slangle formations in several major forms. First and foremost as grains. These grains include bioclasts and apparent peloids, as well as both tabular and rounded intraclasts derived from authigenic apatite crusts or hard grounds. So phosphate does not apparently form the primary cement phase within the stromatolites or surrounding laminated sediments the way that it does in the Brazilian Salitre Formation. Instead, it occurs as these grains bound within the stromatolite laminae or contributing to the interstitial fill. Thin sections further reveal that phosphatic intraclasts may be internally laminated, while some of the more rounded phosphatic grains tend to be more uniformly micro to cryptocrystalline internally, containing diffuse inclusions of organic matter. So these, we think, are more likely to be peloids, or chemical sediments deposited alongside macritic carbonate peloids and sometimes ooids. Phosphatic bioclasts, so bioclastic sediments, take the form largely of internal casts of cavities within small shelled organisms. So the carbonate shells themselves may or may not be preserved along with this cavity-filling cement. But these cavity-filling cements are predominantly composed of microcrystalline apatite, sometimes in these sort of tight mosaics with chert and macritic carbonate cements, and they are often replete with diffuse organic matter, which is the source of some of this, this murky, dark brown appearance that you see. Bioclasts, peloids, intraclasts sometimes co-occur with weakly laminated apatitic crusts, particularly in the Slonigal Formation. So this is solidly in the Cambrian. These maybe sometimes could be argued to have some waviness or crinkliness to them that might suggest the influence of a microbial mat or biofilm on the on kind of the shape of these crusts, but largely they appear to be more like sort of spontaneously forming hard grounds at or just below the sediment water interface, probably. Interestingly, these apatitic internal casts and molds of small shelly fossils, as well as fragments of archaeocyathids, these calcareous cup sponges, those casts have similar mineralogies, internal mineral fabrics, and organic matter content compared to some of these crusts and hard grounds. And both the hard grounds and the casts often cement and include rounded carbonate and silica grains and other carbonate shell fragments, suggesting transportation after the death of the organism and redeposition before this final stage of cementation and occlusion of the internal cavities. So these apatitic cavity-filling cements are well known in these and rocks of similar age and provenance, preserving the internal shapes of small shelly organisms, sometimes referred to in literature as small shelly fauna, small shelly fossils, SSFs, shellies. We'll use them interchangeably here. It has also been well documented that these internal casts form in the interiors of the archaeocyathid sponges. But what is perhaps less frequently discussed in the literature is what appears to be the multiple stages of cavity filling cement, which perhaps speak to the changing fluid chemistry and possibly changing microbial ecology between deposition and cementation and lithification. So here, in these two images, you have the sort of not annotated on the left, annotated on the right. You can see this pore lining carbonate cement. So you had some kind of carbonate nucleation and growth from the walls of pore space, followed then by the filling of this reduced volume, this reduced cavity volume by this organic rich apatite phase. Here is another gorgeous example. This is a fragment of an archaeocyathid cut sponge. You're looking at it in cross section, and you can see you have these cavity lining, nice, sparry, optically clear calcite cements, and then this smaller volume and the inside of those cavities filled with organic rich apatite. So here's another interesting example. And it's interesting to note here that almost the entire cavity volume is filled by these sparry calcite mosaic cements. And then there's this small volume over to the upper left side where you can see this organic rich apatite. And rather than being in the center of the cavities, as you might expect, with a pore filling cement sort of encroaching on this increasingly small pore volume, instead it's sort of concentrated at the bottom. And interestingly, it's concentrated at a bottom that is not necessarily truly the bottom with respect to the orientation of up and gravity at the time of deposition. So that's very interesting as well. Of course, not all archaeocyathid and small shellies contain internal apatitic cements. And indeed, we observed that samples of these archaeocyathid, small shelly fossil hashes that were collected from the centers of these archaeocyath microbialite buildups or bioherms. When we collected samples from the centers and compared those to samples collected from the margins, we saw that the margins tended to have more apatite, the centers tended to be entirely calcitic, but that's a very tentative kind of association, since it's not very often that in exposure you can get the whole true geometry of one of these mounds or bioherms. So what is the difference between these microfacies where phosphate is retained and buried as apatite, and the facies where it is not, or the facies where apatite precipitates in these small shell cavities versus where it precipitates as these laterally extensive crusts? So as with the phosphorites of Brazil, sort of regional, the tens of meters scale depositional setting doesn't really seem to correlate with this variation in the style and extent of phosphogenesis. So what about the geochemistry? So we haven't yet tried to compare stable isotope composition of the different phosphatic and non-phosphatic facies within these Mongolia rocks, the way that we did with the Brazilian samples, in part because of some of the complications with extracting reduced sulfur species and carbonate and apatite associated sulfate in samples that are this rich in organically associated carbon and sulfur. But we have tried another method that I'm pretty excited about, using Raman and fluorescence spectroscopy to look at organic content and some spectral features that may serve as paleo redox proxies. So using Natalia Dritko's T64000 Horriba Raman spectrometer in the Hopkins Department of Physics and Astronomy, we measured point spectra looking at both Raman scattered light and fluorescence emissions using a 514 nanometer green primary laser. So a brief Raman spectra kind of crash course relevant to these samples. I've highlighted here some of the regions of interest in these spectra, including Raman features on the left, which are associated with the vibrational modes of the phosphate ion, carbonate anion, and the carbon-oxygen and carbon-carbon bonds within kerogen. These features are often referred to in the literature as D or disordered and G or orderly or graphitic bands. And then toward the right of the slide is indicated the region of the spectrum where the substituent samarium in the apatite lattice may generate fluorescence bands. Before we go further, I do want to note that the Dritko Lab Raman spectrometer is not optimized for these types of analyses. So there are some features of the spectrum that I request that we ignore for now. So this includes this sinusoidal pattern that you see throughout the continuum. This, I suspect, is due to some specular reflection among some of the optical elements on the breadboard. We should also be cautious in our interpretation of this kind of Bactrian camel hump, which I believe is probably to do with the fluorescence of kerogen. But since we're interested in fluorescence features, unfortunately, it is difficult to try to use a different excitation wavelength to avoid this response. That said, if we zoom in on the Raman active portion of the spectrum, you can see that there's some variation from spectrum to spectrum in the intensity and the center point of the peaks associated with the phosphate and the carbonate. These variations are directly related to variation in the abundance of phosphate and carbonate in the interrogated sample, as well as to the abundance of various substituents of different molecular and atomic radii, which may distort the lattice of the mineral in different ways. Similarly, on the right, the intensity of the D and G bands may be directly related to the abundance of organic matter. So these spectra seem to be telling us that many of the measured phosphatic intraclass, crust, and archaeocyatid cavity-filling cements are all very full of kerogen and apatite, but not all of them. Some crust and cavity-filling measurements contain only carbonate, with apparently variable degrees of magnesium substitution for calcium. And then interestingly, kerogen appears to be more abundant. It's producing a more intense Raman peak in apatitic crusts and interclass derived from the breakup and reworking and transport of those crusts compared to the apatitic cavity filling cements that make up the bioclasts. So very interesting. Now, the fluorescence features are a little bit more complicated. I'll refer those interested to the 2020 papers in Precambrian research by Amanda Garcia and Bill Schopf, in which they explain how samarium substitution in the calcium 2 position of the apatite mineral structure is dependent on the availability of oxygen to bind and distort the lattice to accommodate that samarium cation. The presence of samarium in this redox-sensitive configuration produces variations in the relative size of 2 fluorescence peaks, which at an excitation wavelength of 514 nanometers should appear around 3,000 inverse centimeters. So Schopf and Garcia demonstrated that it may be possible to see changes in the availability of oxygen during apatite nucleation and growth by observing how these fluorescence features change. There we go. Did that go back? me go back. Yes, by observing how these fluorescence features change as you move through different kind of zones of the apatite crystal. But even in their studies, the organic matter does seem to be a problem, peaking as it does right around where these fluorescence features associated with samarium occur. All of that said, there is a measurable difference in the relative size of those two peaks in that 3,000 inverse centimeter region, which could suggest that conditions were more anoxic during the mineralization of those kind of crusts and hard grounds, and therefore in the crust-derived interclass, than conditions were during phosphogenic processes inside these shell cavities. Or maybe not, considering all these complications, but definitely bears further exploring and further work to try to kind of calibrate this method for the interpretation of the Raman spectra. So all in all, this adds up to a very different landscape for phosphogenesis in the biangol and salingol formations of Mongolia compared to the Brazilian phosphorates. This is a landscape that we are still developing as we collect more and better data on paleo-redox conditions and try to do some stable isotope geochemistry that it's texture specific enough to compare microfacies to microfacies. But it's very interesting how different these are when they're ostensibly forming during the same, global episode of phosphogenesis across the boundary. Our study of the Chuluktau Formation in Kazakhstan is still in the most preliminary of phases, and we're working to improve some of both our vertical and lateral spatial resolution of sedimentological and stratigraphic data for these phosphorite deposits of Southern Kazakhstan. But already, just working with our field data and outcrop scale data, we can see that the Chuluktau Formation, which is Cambrian in age, looks quite different from the Ediacaran of either Eastern Brazil or the Ediacaran Cambrian of Southwestern Mongolia. So this is a deeper water depositional environment, and the phosphorite is characterized by this fine granular texture, wherein apatite grains, apparently allochems, were precipitated in the water column above that sediment-water interface, then transported and ultimately cemented with a combination of microcrystalline apatite and silica. Also present there are rip-ups, interclass, and rounded, poorly sorted pebble conglomerates, which appear to all be derived from granular phosphorite deposited elsewhere. And then in a very spatially limited way, there also appear to be some phosphatic oncoids and some small centimeter scale stromatolites, which, in addition to being phosphatic, also appear to be correlated with iron enrichment. Very interesting. And then, of course, as we move up from the Chuluktau into the Shubakti Formation, also lower Cambrian in age, you do see apatitic casts of cavities of small shelly fauna, sort of suspended in this carbonate grainstone as the region moves into shallower water facies. All right. So all of that is a lot to be getting on with. 3 phosphorite deposits straddling the Precambrian-Cambrian boundary. How do they compare? Particularly with respect to potential mechanisms of phosphate delivery, retention, and mineralization in the sediments. So there's some variation among them in depositional setting, though ostensibly all 3 are marine basins during periods of active sort of tectonism and opening of these basins and opening of accommodation space. But in the Brazilian case, you have phosphogenesis being restricted to these digitate stromatolites and associated lamina, and to interclass derived from those stromatolites and lamina. In Mongolia, phosphogenesis occurs within the cavities of calcareous shell-forming organisms, including archaeocyathid cup sponges in this range of different millimeter to 100 micrometer scale small shelly fossils. Phosphogenesis also in Mongolia occurs as crusts in hard grounds, cementing together both phosphatic and non-phosphatic grains, including allochems that may have precipitated from the water column and bioclasts. In contrast, phosphogenesis appears to have occurred primarily in the water column in Kazakhstan, with phosphate being buried as these chemical sediments, and maybe in some like spatially restricted cases in these microbially mediated oncoids. Oh, and I left it out here, but Kazakhstan also has some of these phosphatic internal casts of small shellies, though they're a little bit rarer. Now, directly comparable geochemical data sets that are texturally specific to phosphatic microfacies, all the different phosphatic microfacies, are not yet available for the Brazilian, Mongolian, Kazakh phosphorites. So we haven't made the same measurements and the same with the same analytical conditions in all of them. And that's something that we're working toward. But the data we do have suggest in all cases that fluctuating redox conditions are associated with the phosphorites, and therefore potentially maybe microbial metabolisms suited to operate under those fluctuating or transitional redox conditions. Additionally, it is worth noting that none of these phosphorites contain particularly well-preserved apatite permineralized microbial fossils or fossilized animal tissues. In contrast to that beautiful preservation you see in the Ediacaran and Cambrian formation, or at least in the more shallow water facies of that formation. So, though, despite the lack of fossils, all of them are also do contain elevated kerogen. So there's a lot of diffuse organic material associated with the phosphatic microfacies, and more so in the phosphatic microfacies than in the non-phosphatic. So all of these things together, what controls phosphorus mineralization? What controls it at these fine spatial scales, both within and among these studied phosphorite deposits? So we can rule out, I think, variation with respect to the delivery of these iron oxide phosphate complexes to sediment. We don't see a lot of sedimentological and geochemical evidence for that. We also don't see that much variation with respect to the apparent availability of calcium or fluoride in the earliest preserved textures. But the preserved, spoilers, but the preserved mineralogical and geochemical features that we do have seem to suggest some variation with respect to the amount or the type of associated biomass, and possibly then the types of metabolisms in operation at the time of phosphorus mineralization. So you have this variation on these tiny spatial scales. The only thing that seems to vary on the spatial scale of centimeters to millimeters plausibly is microbiology and the effect of different microbial communities on the pore fluid chemistry from one little cubic centimeter of pore volume to the next. So these observations, our interpretation thereof, seem to support this hypothesis, which is grounded in theory, experiment, and observations of modern phosphogenic environments, that phosphogenesis is locally controlled by excess P sequestration and release to pore fluids, facilitated by microorganisms living under transitional or fluctuating redox conditions, conditions which themselves are affected or created by these global scale environmental changes, a la those discussed by Tom Loxo and collaborators. But this kind of begs the question of why should phosphorus accumulating microorganisms be concentrated in specific types of structures, particularly biological structures, like these specific stromatolite morphologies or these kind of crinkly crusts, or in small shelly and archaeocyathid cavities. If it's the distribution of these phosphorus accumulating biomass that's controlling it, it's like, well, what's controlling the distribution of the phosphorus accumulating biomass? So one possible explanation may just be the taphonomic window. This is the explanation favored by Jessica Krevling and collaborators in their paper on the taphonomy of phosphatic small shellies, in which they observed that only the smallest size fraction of these small shelly fossils had these apatitic cavity-filling cements. However, this phenomenon has not yet been demonstrated in the archaeocyathids, nor does it seem to apply to other phosphatic microfacies, such as the crusts and the crinkly or wrinkly laminae or the stromatolites. We don't see a lot of textural evidence that there were variations in pore space and connectivity between the phosphatic and non-phosphatic facies there. We're running out of time, so I'll roll through this a little bit. But another possible explanation may just be that organic matter has phosphorus. Organic matter is abundant in microbial mats and the decaying bodies of tiny animals inside of their shells. But something has to raise the phosphorus content of the material inside of those shells and inside of those cavities. So it doesn't seem like typical biomass attracting heterotrophic microorganisms after deposition and burial could necessarily explain the amount of phosphorus necessary to fill those pore volumes that we're seeing. So I'm going to skip ahead here. Everyone knows about ATP, its use as energy currency in the cell, but it turns out that ATP requires an active metabolism to manufacture by the cell. And so one strategy that certain microorganisms have for dealing with metabolic stress is to accumulate polyphosphates. So polyphosphates, like ATP, have these high energy bonds between these orthophosphate groups. They accumulate chains of these several thousand orthophosphate groups all linked to one another. And in times of metabolic stress where they can't manufacture ATP the way that they were, they rely on the dephosphorylation of those polyphosphate chains. And indeed, this is a phenomenon observed in a range of microorganisms, which also, interestingly, are particularly abundant in modern phosphogenetic settings. Moving on from there. So why would phosphorus accumulating, polyphosphate metabolizing microorganisms want to be in these particular settings? And so this is kind of the new idea that I'm just going to tease out there before we open things up to questions. I think potentially the accumulation of polyphosphate by these microbes could have been indirectly useful to multicellular organisms that they lived in spatial association with. So could it be possible that phosphorites deposited across the Precambrian-Cambrian boundary, and particularly these styles of phosphogenesis that occur in association with bioclast? Could it be that this is telling us something about the evolution of a symbiotic life strategy? This is a well-documented phenomenon in modern organisms. Modern animals don't have polyphosphate accumulating metabolisms. And indeed, modern sponges benefit from these symbiotic associations with polyphosphate accumulating organisms, which provide them with provide the sponge with a food source, and in return, perhaps take advantage of the unique kind of microenvironments created inside of these organisms, shell cavities, to provide them with a steady flux of essential nutrients. So phosphorites, as potentially a record of this evolutionary strategy for dealing with fluctuating or transitional kind of redox conditions. An idea. All right, I'm going to stop it there, and I think we can go to questions.
All right, thanks, Cecilia. Really cool stuff. We'll go ahead, yes, and switch to the question and discussion. You can ask a couple ways. You can type your question into the chat and I can ask it for you, or you can find the reactions button at the bottom of your screen. Click that, and the raise hand is that first button at the very bottom. So I'll give a few moments to see that.
Maybe I can start.
You want to start?
A quick question, maybe a clarification. So symbiosis would imply that this relationship were in lifetime. And do you see in petrography evidence that a phosphate formed during the life cycle instead of like after animal or fossil animal was dead and sort of phosphatization started?
Yeah, so this is a great question, and this is sort of one of the main things that I've been thinking about obsessively in the last couple of weeks. When we're looking at these rocks, we're looking at, this time integration, and so any phosphorus enriched biomass enclosed within bacterial cells that were associated with these living organisms, when those animals died, and those microbes presumably either died with them or went on to like scavenge all of that material is then going to be oxidized and remineralized by microbes living in pore sediments. And so it seems to me that and especially because they're so phosphorus rich, they're such an attractive target for heterotrophy. So I think you'd be unlikely to preserve this lifelike association between the bacteria and animal tissues necessarily, unless they're, it's sort of preserved in this more abstracted way in the stable isotope record, say of like carbon and oxygen. But what I think we can see is how the apparent distribution of phosphorus kind of tells us a story about when this phosphorus got stuck there. So the question would be, it's like, well, why would these polyphosphate accumulating organisms want to congregate around a certain type of animal, unless that animal was that animal and any associated microbiome from that animal were particularly phosphorus enriched. So it's tricky and requires this like really kind of this comparative analysis of, okay, where is the phosphorus, and is it does it seem that it was especially likely to have like phosphogenesis happening within certain types of organisms, or is it more a matter of these like post-depositional conditions?
Yeah, I guess like if you have this animal with tissues, it could create like a redox, micro redox environments that would be good for the sulfur oxidizing bacteria. But I wonder, you can maybe do like calculations, like sort of how much of this sulfur oxidizing bacteria need to be in this organism already, but during post-mortem you kind of just use this phosphorus to form phosphatization versus you need external source of phosphorus.
Yeah, I think experiments would be really powerful, alongside these sort of calculations and like models. What I need is a collaborator who already knows how to grow sponges in an aquarium because I do not. But there's some incredible literature out there about the kind of physiology and microbiome and associated microbiome of modern sponges that like show that most of the phosphorus indeed in a sponge is actually not in the sponge's tissues itself, but in these bacterial symbionts associated with the sponge. But I would love them to, lend me their phosphorus enriched sponges, so that I can kill them and bury them and see what happens.
Or you can just think like, okay, like in sulfur oxidizing bacteria, how much phosphorus could be. So sort of like how many of them you need to put into animal, but where decay would result in this phosphorus content.
Mm-hmm.
Great. So I may be letting people talk, and I have a few more questions for you.
Okay, yeah, we got four messages in the chat in that time, so let's go ahead. Catherine Giles asks, or says, interesting idea on symbiotic relationships. Do we see organisms with phosphatic shells, like brachiopods, copepods, having symbiotic relationships?
Yeah, with phosphatic shells. So I am not I've been very firmly in the Precambrian until very recently, like branching into Mongolia and Kazakhstan. And so I'm not terribly familiar with literature on like phosphatic shell forming like modern organisms and their symbiotic relationships. We do see in these old rocks, we do see some phosphatic shells. It appears to be, it's like patchy and it's rare, like mostly, and it seems to be more a replacement feature, possibly a result of calcareous shells getting deposited these in these settings where you have like formation of like phosphatic crusts and hard grounds, maybe. But I haven't seen a lot of examples in these rocks of primary apatite shell forming in these. Yeah, like at least not in the ones that I've seen. But that would be. But yeah, but certainly like as you move into like the further into like the Paleozoic, like there are tons of phosphate shell forming organisms. And yeah, I think that's a super interesting idea.
Cool.
Would be interesting to know whether or not, their relationship with these organisms is like, key to the evolution of like phosphatic biomineralization.
All right. Thanks. On to I have one here from Brian Pratt. Yes, in the Mongolian samples, some phosphorus grains are black and some are brown. Is this due to the relative proportion of organic carbon? At least one dark grain seemed to contain tangled filaments.
Yeah, so let's go back to those. I love nothing more than just staring at thin sections with people because everyone I think focuses in on and notices different things based on samples they've looked at before. So let me find one of those grainy ones. Boop, 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 boop. Here we go. Yeah. So in these images, there's a jumble of micritic rounded like carbonate grains alongside these phosphatic grains. And generally in these plane polarized photomicrographs taken under plane polarized light, the phosphate ends up looking brown because of the presence of all of this like kind of diffuse kerogen. And where you see like the truly black or the truly opaque, if you like go into higher magnifications, it can sometimes seem it's like, okay, it looks like it's just like particularly murky and it's got like a lot of these inclusions. And sometimes it's clear that there's secondary phases like opaque, like iron oxides, or sometimes would appear to be oxidized like pseudomorphs after possibly pyrite. And so, yeah, so that like kind of like dark color like somewhat depends. Though I do agree with you that there are definitely some sort of like kind of filamentous or like somewhat like disorderly like striated like textures. I had some slides about like putative microfossils that I ended up cutting for time. But they are, so yeah, I think that generally the organic matter is like pretty diffuse in these things, but sometimes there are clumps. And in those clumps, like you can see sort of faint textures that could be the result of that organic matter sort of being concentrated in individual cell bodies. But they don't make for particularly impressive fossils because of the sort of lack of morphological information contained there. But yeah, I think that like one could pore over these and pore over some of these samples and maybe find some more like compelling filamentous and coccoidal structures. And certainly there's lots of beautifully preserved microfossils in the chert facies of in Mongolia and in Kazakhstan. But yeah, but not in the phosphorite, interestingly.
All right. The next one is from Liu Yi Lu. It says, thanks for the talk. Does phosphorite always deposit in high energy environments or experience transportation?
So no, not necessarily. So in the case of the Kazakh phosphorites, I don't have the thin section images yet to show you because just got those back. But it seems like it was a deeper water, less energetic facies, like mostly planar lamination, much finer grain than the stuff that we were seeing in Mongolia and in Brazil. And it seems like those grains, rather than being, they could be the result of kind of like a winnowing and reworking effect, where non-phosphatic grains were transported away and you kind of concentrated the phosphatic grains left behind. But they are so fine grained. I I would be, or vice versa, phosphate transported away and then deposited in these deeper water environments. I think, yeah, you'd need the thin section. You need to look at them in thin section, I think, to try to look for internal textures that were maybe indicative of like the ripping up or reworking of like laminated crusts versus like kind of nucleation and growth of apatite crystals sort of omnidirectionally around like a nucleus, like you would expect in the water column. But yeah, you can, and of course, like, the Geoshodantoa formation and other phosphorites, they've shown there's, different styles and qualities of like phosphorite mineralization in the shallow versus like the deep water facies. And it seems like the shallow water facies are maybe more conducive to like preservation of some of those beautiful apatitic microfossils compared to the deep water facies. So yeah, all this to say, it doesn't have to be in a shallow water environment. And likely it probably depends on like where that chemocline is located. And because that sort of transitional zone seems to be where phosphogenesis occurs. And that can happen in the water column, it can happen in sediments, more likely to happen down in the sediments if you're in this highly energetic setting and you're mixing a lot of, and it's fairly oxic, like all the way down to the interface. But yeah.
All right. The next one is from Jackson DeHorn. He has a lot of questions. Are you able to open the chat? And
let's
it might be
see.
easiest if you kind of read them yourself and address them as you go.
I should be able to. Haha, I found it.
Okay, great. If I can ask, that you read them aloud for those watching on YouTube.
Yes. All right.
Awesome.
There's Catherine Giles. There's Liu Yi, and then okay, Jackson. So Jackson says, I am curious if you look at these thin sections under using reflected light microscopy to see if any sulfide minerals are present in the organic kerogen-rich zones. Thinking about this, because if you had sulfides precipitating and being weathered, this would generate acids, especially with increased sulfate and hydrogen sulfide with deposition. That could locally cause pH fluctuations that would be more conducive to aid in precipitation of the apatite-rich cements. Hence, couldn't the iron be more important in incorporating in surface complexation processes in the pore spaces? No, yeah, they apologize for having lots of questions. Don't apologize. These are awesome speculations. Okay, yeah, so I do look at them under reflected light. And generally speaking, so I was particularly interested in looking at them under reflected light when I was trying to pick targets for sulfur isotope geochemistry in the with my Brazil rocks. And it's not that iron sulfides were rare so much as just sort of very small and very, kind of diffuse and spread out. So they weren't there wasn't sort of like a unique enrichment in like iron sulfides or pseudomorphs thereafter associated with the phosphatic facies. Similarly, in the Mongolian samples, you see some iron sulfides and pseudomorphs thereafter, but they're not kind of uniquely concentrated with or uniquely associated with the phosphatic compared to the non-phosphatic phases. But yeah, so that's very interesting and could be, as you discussed, sort of the result of this like these diagenetic processes that sort of change the primary mineralogy and therefore the chemistry of these pore fluids under increasingly closed system conditions as they get buried deeper and deeper. So let's see, to your next question. But yeah, you'd think that that would certainly, yeah, those processes would certainly affect pH on kind of a highly local scale from like pore to pore. So the question, sort of becomes, it's like, okay, well, were there iron sulfides that, that then went away and like weren't buried? Why weren't they buried if like conditions were relatively like anoxic? It's I think, yeah, I think that it's definitely important. The relationship between biogeochemical cycling of sulfur and phosphogenesis is super important, and coupled definitely at these like very highly local scales. I just also think that microorganisms are responsible for sort of pushing that sulfur around between reduced and oxidized phases, and controlling or responding to these pH changes.
All right. Cool. Next, we have one from Karthik Sender Sarwan, who says, found the hypothesis and talk very interesting. Is there any quantitative evidence concerned with the phosphorites and phosphogenesis that may be useful in determining the degree of symbiosis, fluctuative or obligate? And does modern microbe-mediated phosphogenesis preferentiate between one nuclide of phosphate over the other? And if such preference exists, how would that affect phosphogenesis in the Precambrian-Cambrian transition?
Cool, awesome. These are awesome questions. So I don't know yet what a quantitative evidence would look like for determining facultative versus obligate symbiosis. Other than, I guess it's semi-quantitative, but like other than sort of like the number of instances of like true correlation between various paleo-redox and paleo-ecological proxies that we get from geochemistry, and like and then like the textures that we see, the textures that are biological versus abiological in origin and things like that. So like looking systematically for those correlations in different types of phosphorites, I think could provide could quantitatively, I think, support or not support the idea that symbiosis is necessary as opposed to just decay after the fact and after deposition. In terms of determining the difference between facultative versus obligate, yeah, again, you'd kind of look for the occurrence of it's like maybe in environments where you see geochemical evidence of highly anoxic, stressful to aerobes redox conditions. Maybe if in those environments you only see apatitic or internal apatite casts of maybe cup sponges, and then in environments where you have evidence of a more oxic setting, you get maybe a mix of apatitic and calcitic, or just calcitic archaeocysts, and you see that trend bear out over and over again in all the examples that you look at. Then like maybe you could have like this, you could make an argument based on like that quantitative information. It's like, obviously, in order to do quantitative work, like you need lots and lots of rock. But yeah, I think potentially there are, maybe you could determine facultative versus obligate, just like based on the frequency of occurrence of these associations. And then your other question was, in modern microbially mediated phosphogenesis, do the phosphate accumulating microbes seem to prefer to stick one nuclide or another onto the phosphate? So tentatively, I will say yes, but because I've certainly encountered in the literature the prevalence of like GDP and GTP, or like, so that's like guanine stuck to the phosphate chain via like ribulose, as opposed to like the adenine stuck to the ribulose, stuck to the phosphate. And that's something that like doesn't tend to occur. It doesn't tend to occur in animals, but I don't know how widespread or like universal that preference is among polyphosphate among microbes that like utilize polyphosphate metabolisms. But yes, that is a very interesting point, and maybe something that could leave a geochemical signature, which we love. But yeah, okay. And then let's see. Are there any more that we missed over here?
I think we're good in the chat. I was going to ask one when we didn't have another. But what I was just more curious, I was curious about sort of the phosphorus to volume ratio of organic matter versus
Yeah,
CFA.
let's see. Yes. So let's go there. There's a little kind of back of the envelope calculation presented in Jessica Kreveling's paper on phosphogenesis in small shellies, where they make the argument that the volume of, so you take the internal volume of one of the, ooh, I went all the way over here, but maybe I should have gone back here.
I saw this,
you take,
you had 1% and 16% there.
yeah, so that's the mass fraction. I just wanted to pull up one of these small shellies. Boop, 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 boop. Here we go. Okay, so if you have, say, like a tubular, like small shelly, you can like get a rough estimate of its internal volume. You say if you fill this entire internal volume with biomass of kind of a standard like Redfield ratio, and like make some assumptions about the density kind of that biomass and how much of it is, water versus how much of it is like truly organic compounds and other stuff, then you say, okay, this, volume can only contain this much phosphorus, based on that assumption, with a sort of phosphorus mass fraction of like 1%. If you fill that same volume with apatite, accounting for, subtle variations in the density due to like different quantities of different substituents, et cetera, you get kind of a mass fraction of 16%. So the idea is that, okay, standard biomass, with all these kind of standard assumptions, isn't enough to provide all of this phosphorus. So presumably you need either a higher mass fraction of phosphorus in like the primary, and like the body of the organism that originally occupied that, and that could be due to, presence of phosphate accumulating bacterial symbionts, for instance, or it might be due to the transport of extra phosphorus in there by heterotrophic microorganisms that are going into that volume, and oxidizing the organic matter there, remineralizing it, releasing, releasing more phosphorus. And then you'd have some kind of control on, diffusive transport of the free phosphate out of that space. Like you have to keep it there and store it there somehow. And that could be either by like the spontaneous precipitation of apatite cements, or it could have something to do with the morphology of the shell itself, sort of limiting the exchange between the fluids inside the shell and outside the shell. So this is actually an experiment that I'm setting up right now, looking at influence of like shell volume and morphology experimentally on like the sort of the rapidity and extent of like precipitation of like apatite, because I think it's maybe kind of a non-trivial problem. Like obviously like smaller volume, more closed system, easier to shove the saturation state of apatite versus carbonate around with relatively few microbes. But yeah, like you still have to deliver extra phosphorus there. It's not just about retaining the phosphorus that was already contained within that living organism. You have to deliver more stuff there.
By 16 times as much effectively.
Yeah.
So I'm wondering, like, if we can, it would be interesting to see, like, all the different pathways, like taking what is the average, phosphorus scavenge rate by oxyhydroxide minerals, and how much of the oxyhydroxide minerals would you have to deliver and then reduce to actually achieve this versus other mechanisms. I'm sure you could kind of do some calculations to figure out which one would have the most demanding and versus most realistic process and stuff. I don't know. That I just kept thinking about that through your
No,
talk.
yeah. I think that, it should be a relatively straightforward calculation to make. It's just about, okay, how do we, where, what will all of our assumptions be in order to make that model work? Like, for example, with like the iron oxide adsorption scavenging of phosphate, then you have to transport that into like the limited volume and reduce it. And then something has to happen to the iron. And the iron itself can also be a common substituent in apatite as well. And the presence or absence of iron, like, depending on like, pH conditions can also affect, can also kind of create like a kinetic barrier to precipitation of apatite and stuff. So I think, but yeah, I think it's the basic calculation of like, okay, how much iron would you need given the amount that we expect to adsorb to the surface of a grain, like of this size over this time period. And then, what effect would like the release of that like free like ferrous iron into solution have on apatite saturation and like precipitation. And so, yeah, someone should do that. Should it be me? I don't know.
Not enough time in life. Okay, before I let Andre get on, I'll ask one more. I forgot how to pronounce your third phosphorite already, but you talked about iron enrichment. What phase is that iron
The
in?
tulip tau. That is an excellent question. I like super glazed over it because they're so weird and wonderful, and I'm really excited. To look at them in thin section. But just doing XRD of just like kind of bulk like powdered like admixture, it appears to be hematite mostly. But that's only that's one sample. One sample all mixed together. And so, yeah, I'm interested to see like when I actually look at it in thin section and get some Raman maps and maybe do like some EDS mapping and estimates as well, like I think, yeah, it'll be interesting to know like how kind of uniform that is.
Yeah. And I'd love to see your oncolytic lamina in EDS spectra.
Yeah. Oh, I bet they're going to be so beautiful. They look very beautiful, like visually. Just yeah, looking at them in outcrop and in like I've done some like polished like slabs and sections. But yeah, they're like, so much of it is going to be totally opaque. It's like looking under the
Yeah.
microscope. But yeah, EDS, uh, XRF, Raman maps, like yeah,
Mhm.
I bet there's a lot of cool structure in there.
Cool. Good luck with it all. Go ahead, Andre.
Cool.
Yeah, I have a couple questions. So one, um, I was always interested in distribution of phosphates through time, and particularly for Neoproterozoic ones, like when do they start? Like, so you showed some diagrams from 70s where kind of generally we accept it's Ediacaran to early Cambrian, but I'm wondering if you or somebody else has any compilation, sort of like how far in Ediacaran can you push it, or even if it's extend to Cryogenian or not.
Uh, oh, I think this
Yeah, that's
You got a
kind of
swap display.
critical age.
there it
Yeah,
goes, a little bit bigger. Yeah. So I'm fond of the slightly more old-fashioned, like kind of hand-drawn plots that like look a little bit crispy. Like, I don't know, it like really emphasizes that like this is a general idea that we have. But there are more recent papers that try to like refine this compilation stuff, but I haven't found one yet that has really tried to be kind of quantitative about it, um, or more quantitative about it, and sort of estimated like the relative kind of size, the amount of phosphate, uh, or like put some constraints on like the amount of phosphate represented by like different represented in different formations, and you know put that on a map, put the plot next to the map and everything. um, that said, like the stuff that I have seen seems to suggest that there are older phosphorites like mesoproterozoic, uh, at least, um, but perhaps not as big. And not as big is also kind of an interesting designation because I feel like that kind of comes from the economic geology literature, where like big means economically important because it's particularly concentrated, or like, or like whatever phase the phosphate is in, whatever form of apatite is like really dense, rather than sort of like diffuse and associated with these other things. So I think it's quite possible that, there's other episodes of like phosphogenesis locally, like, and globally, and that go back like earlier, but that are just, they're not as economically important, and therefore, maybe they haven't got as like close a look, and maybe so that they're like underreported for that reason. um but yeah, but I have seen there are phosphogenic episodes,
I guess
like earlier.
what I was interested in, I mean, there is polyproterozoic episodes and there is like 1.7, but what I was interested, how far back Neopret, or let's say the current Cambrian goes, because
Mm,
as you mentioned, people try to link it to ocean oxidation, and so would be interesting
yeah,
see like how close temporary it's fits. And this diagram kind of okay, but age constraints change so much from 79, and not to mention Precambrian Cambrian boundaries. So it's really
yeah,
would be interesting to update it with new ages and And specifically, you mentioned that phosphorites might not be related to phosphorus flux. I'm not sure how well do we know it, because like if you look in Phanerozoic, I mean, certainly ocean was oxidized, but still there are these periods of phosphogenesis and periods with not much of phosphate. So
Yeah,
must be something else controlling.
very interesting. Yeah. I see what you're, sorry, I misunderstood. I see what you're saying about like the Neoproterozoic, like how early, how, Precambrian, Cambrian transition, post-glacial overturn, oxygenation of the oceans, like all of these, there's a little bit of, hand waviness when we talk about them in the general, but like specifically, it's like, we know that oxygenation is like happening. Oxygenation of the oceans is happening. And I think becoming like generally widely accepted that rather than like a big single step on like a short time scale, you probably have this like gradual and like spatially heterogeneous rise of oxic surface waters, and oxic to oxic deep waters, as early as, probably starting somewhere in the like somewhere I don't want people to get mad at me, so like I don't want to say definitively in like Paleoproterozoic, but it's but we know like there's evidence of like patchy oxygenation and stuff, and that it's you know going back that far, and then things ramp up. So it's I think that like the time scale of like oxygenation is maybe long of gradual oxygenation is like maybe long enough that you could see like argue that it's like, okay, like the onset of this like phosphogenetic episode or whatever like post dates that, but like post dates like the beginning of that. But like with respect to like orogenies and like input of like detrital appetite from the continents and things, like those are things happening on like shorter time scales with like tighter, very much tighter like age constraints and things. And so I really enjoyed that Loxo paper. But yeah, maybe it doesn't like completely rule out these like secular changes in like external phosphorus flux.
Yeah, it would be interesting to see like sort of when phosphogenesis started even in Ediacaran, and how does it correspond to other events like oxygenesis or orogenesis or anything else. And another question, I wonder if you thought about sort of once you get some precipitation of phosphate, like some template, it would be much easier for phosphate to continue to grow, like basically like by phosphate ripening. So you might not necessarily need to have all this phosphate formed from what was during lifetime present in a fossil. But as long as you form some crystals, they will continue to grow post-mortem by diffusive flow. And to form this template, I wonder if the composition of the shell, if it's aragonite, calcite, would it play any role? Is there any kind of similarity between apatite and calcite versus aragonite that would enhance phosphate deposition or not? I don't know if you have any comments or thoughts on it.
Yeah. So the idea of post-depositional and possibly post-lithification enrichment of phosphate, and is something that we in like largely like try to investigate like texturally and mineralogically. And like texturally, like it doesn't seem like there's a ton of evidence for like, you'd have to have like this fluid carrying a bunch of phosphate, possibly, maybe meteoric fluids carrying remineralized organic matter from like the surface down into the rocks. You'd have to get a fairly large amount of water into the rock. Like, and like we don't see a lot of textures like consistent with that. But of course, like it's also like the mobility of phosphorus between like fluids and apatite phases is like also very dependent on like temperature and pressure and the nature of like the arrangement of like other phases and things. So like it could be possible. I feel fairly confident that like the phosphate enrichment like in the rocks that we've looked at like seems like a primary or early diagenetic signal rather than like a later like late stage like diagenetic signal. But
Yeah.
yes, like sorry. So that was the first part.
Yeah, I didn't
Sorry,
mean
go
post date very late, but I meant but once you have a template or once you have a little crystal there, whatever fluid circulation happened during early diagenesis, it would sort of tend to grow around it rather than
Yes, yeah.
yeah. And
Sure. Oh, okay. I see what you're saying. So you're talking about like, early diagenetic, you have a
very
little bit of
early,
apatite
yeah.
crystallization, and then you have this somehow like this transport of phosphate enriched fluids, like they're like moving through the sediments, and the presence of like a little like nucleating crystal itself might be enough to like create this little nexus for like phosphate accumulation and enrichment. I think that's probably a calculation that could be made like based on some experiment, like based on like empirical data about like phosphate crystal growth under like low temperature conditions and stuff, because it seems, like if that nucleation, because but that aside, like, yeah, I'd want to know like what is that nucleation point, and is it like facilitated by like some complexing between like free phosphate and organic matter, or is it like just like the physical surface like of the shell? And in that case, like why this shell and not like that shell over there? Like, yeah, because it seems like more than random. Um, like that there's like that there is like pattern to it. It's
Yeah.
very interesting. Yeah.
And the last kind of not just a piece of information. Sorry for taking too long, and
No.
a couple more questions in chat room. But we work on Baltica on the latest Ediacaran, and we have a lot of phosphate nodules you mentioned in your introduction. So this phosphate nodule, basically like a fist size or even larger, like 10 centimeters in diameter. And we see it in shales, but these shales are not very organic rich. We maybe have 0.3, maximum 0.7%. And so this balls of phosphate developed in the sediment. So it's another example of Ediacaran phosphate formation. So if you
Cool.
have any interest, we can chat it later, and you're welcome sort of
Yeah,
to look and work on that.
cool. Yeah, I'm fascinated by like phosphatic concretions and stuff, and I've mostly been looking at like phosphatic concretions from much more recent, like like Miocene, like lake sediments and like stuff from like the Jurassic and stuff. Like I would love to, yeah, that's a whole different, a whole different game, but similar, maybe similar chemistry happening. That's very interesting.
And they have kind of radial textures to them, and they have, like, for some reason, galena in the
Ooh,
center of them, so we kind of figuring out what can be done with them. Okay,
yeah,
thanks.
really cool.
Yeah, maybe we can chat more.
Okay.
Awesome.
Jackson asks, do you know what the clay minerals are in these samples, if any?
Yeah.
Yeah.
So not that many clays, except, well, certainly not, almost no clays in the Brazilian, in the Salitre Formation phosphorite. In the Mongolian phosphorites, you sometimes see glauconite, and that glauconite is sometimes very closely spatially associated uh, with the like apatitic crusts. And there are also some like glauconitic, uh, small shelly fossils, and occasionally like archaeocyath fragments as well. Sarah Pruss has a paper with some really beautiful images of that. And largely like the presence of it appears to be like authigenic glauconite. So not a lot of detrital clays, but like authigenic precipitation of glauconite, which, because it has mixed valence. It contains like mixed valence iron, iron 2 and iron 3. It's sort of seen as more geochemical evidence for kind of fluctuating or transitional redox conditions, precipitation happening at that oxic-anoxic boundary. But other than that, not many clays, no.
All right.
Sorry. And in Kazakhstan, oh, sorry, no, not in Kazakhstan. In the lower part, like the Ediacaran part, there are some very glauconite-rich shales that have previously been reported as phosphatic shales, but if they do have phosphate, it's like below like the detection limit of the XRD that I'm using. Glauconite seems to be the dominant phase in those shales alongside quartz.
Okay. He adds that point, or that the reason he's wondering was because about Archean origin of life, using clays and hydroxyapatite as the source for phosphorates to promote RNA polymerization.
Yeah,
So just
super
some back.
interesting. Yeah, clays, I think, are slept on as participants in biogeochemistry at like these really fine spatial scales and stuff. Or at least like slept on in a lot of the like kind of phosphorite biogeochemistry like literature. But yeah, good question.
All right, there's nothing else here, and we're coming up on 2 hours, which is the longest I think the longest one we've had in a long time. A lot of really good discussion, a lot of good questions. Thanks so much for your time, Cecilia, and really cool stuff.
Yeah, this is really awesome. Thanks for having me.
Yeah.
And,
okay. Great. Well,
we have a next talk by Adolf Janke from Weber University, but I guess Alex will send it later this week.
All right. Great. See you,
All right.
everybody. Take care.
Cheers,
Thanks.
y'all.
Mhm.